Hello my dear amazing learners, welcome back to Free Hit. I am Nisha, University Gold Medalist, your biology faculty. So today we'll be discussing about the male reproductive structure and the pre-fertilization event, because the male reproductive structure has to be formed before reproduction. First we'll understand the structure and then we'll see how they are formed. Fine. Now let's get into the structure. I prefer study the diagrams, understand how they look, how they are. It will be very easy to answer questions. Okay, so just now, in the previous video we saw this complete structure. What did I call them as? Andrecium, okay, or I may call them as stamen. Okay, I'll be telling you all the points in the diagram itself, and then we'll look at the points. Okay, so first try to put a lot of attention in the slides where the images are kept, because it will be like giving you an overall picture, and then we'll look into the points also. Don't worry. So this is anther. This is filament. Okay, so the topmost portion is anther. It is usually free. Remember, it is usually free, but sometimes they may also be attached to either gynecium or some other port, but they are usually free. This basal portion, it is attached. Okay, the basal portion is attached to, it may be attached to a petal. In that case, we call them as epipetalis stamen. Okay, or they may be attached to thalamus. Okay, so of these two, this is most common, attaching themselves to thalamus. Okay, so the top portion is usually free, the bottom portion is usually attached to petal. If it is petal, epipetalis. If not, it is thalamus. Okay, and then when you look at the anther, you can clearly see this outline. This is one, this is another. So anther is bilobed. Okay, and then I take only this, and if I look into it, I can find, so they have given this, no, there's a longitudinal groove which cuts it lengthwise. Okay, this is theca. Understand, the complete structure of anther is divided into two lobes. Okay, that is I have written as bilobe, and if I take only one lobe, we have theca. So in one lobe, how many theca? Two. So most of the anther, angiospermic anther, are dithecous, but still there are monothecous anther. Fine. Okay, so just come up with a basic math. In anther, we have two lobes, and in each lobe we have two theca. So how many lobes do we have in entire anther? So two lobes. How many theca? 2 into 2, 4 theca. Okay, just remember, an anther has two lobes and 4 theca overall. And if they ask theca per lobe, it is 2. Okay. Now, this is very prominent here. Look at this image. So this longitudinal grid. Okay. This complete stuff is one lobe. Okay, and this one is another lobe, and you can see this is one theca and this is another theca. So how many thecas you have? 1, 2, 3, 4. And inside theca, what do we have? So this, no, the round I have drawn. So that stuff is pollen sac. So sac is something that is used for storing. So what it is going to store? Pollen grain. Okay. The line that we drew here is this line of dehiscence. What is line of dehiscence? Line of breaking. So the point where this pollen sac will break. Okay, it just splits into two and causes the dispersal of pollen grain. Okay, so imagine we have two lobes and each lobe has two theca. Theca is separated by line of dehiscence. This line of dehiscence opens and it liberates or it disperses the pollen grain stored within it. So this is very important. Pay some attention to it. Okay. And then one more important point. This length may not be same in all organisms. For different species, length of filament and number of filament are different. Okay, the number and length of filament may not be same for all species. So different, some of them will be having this much filament length, some will be having this, some will be having 10 filaments, some will be having only one. There are varieties. Okay, let me have a quick revision of this and we'll move. So the complete structure is known as the androecium or stamen. The top portion is free, it's known as anther. The bottom is attached to petal or thalamus and it is known as filament. Filament may be of different length and different numbers in different species. Okay, and then this anther has two lobes and each lobe will have two theca, and the line on the center is known as the line of dehiscence. Line of dehiscence breaks and it liberates the pollen grain stored in the pollen sac. Okay, and anther has two lobes, each lobe has two theca, so an anther has four theca. So we call that type of anther is dithecous. Dithecous refers to this condition. Bilobed refers to this condition. Okay. So, and another thing, they'll call this proximal end and this is distal end. Fine. Proximal, distal. These are the terminologies that we commonly use to understand. Just a minute, I think. Yeah, I do have it. So this proximal end is being attached to the petal or thalamus. Fine. So if this diagram is okay, the theory is like a revising part. It will be revising. Just make sure at the end of this video you must be able to recollect most of the points. If you are not able to recollect, then there is some problem from your side. You are not paying proper attention. If you pay proper attention, then yes, these things should easily be coming up in your hands. This is very simple. With the diagram you can understand. So stamen has two parts, just like we told. One is long, slender. What is slender? It is like flexible. Aren't filaments flexible? So they are flexible. Because they are exposed to air, let's imagine they are flexible, they are hard, rigid like this. If a very high speed wind comes, they break. Okay, we don't want a filament break. So it is flexible, so just goes and comes back. Okay, it is long, slender, stalk is like an axis. It is called filament, and the terminal and double underline, it is generally bilobe, but sometimes it may also be single lobe. Generally bilobe. And longitudinal groove, we told you on the line of dehiscence. It separates theca in lengthwise. What do you mean by lengthwise? So if it is in anther, it divides like this. It doesn't divide it like that. So this is lengthwise. And a typical angiosperm anther is bilobe, with each lobe having two theca. Didn't we discuss this? Dithecous. The anther is four-sided. Okay. This is the anther. We discussed they will be having two lobes. Each lobe has two theca. This is from this view. Let me look at this from the top view. How it is going to look. Okay. So, give me a minute. You have to look at this. Yeah. Just look at this. This is from the top view. You can see 1, 2, 3, 4. It's like tetragonal. Okay. Like for example, if you say square, it must be like this. If it is rectangle, it must be like this. Rhombus, something. But see this, side 1, side 2, side 3 and side 4. It's not like proper, it looks like a trapezium, but it's not actually trapezium. But how many sides does it have? 1, 2, 3, 4. So what can we call tetragonal, right? We call it as tetragonal. And there is four-sided tetragonal consisting of 4 microsporangia, and it is pollen sacs. They mean pollen sacs by microsporangia located at the corners 2 in each lobe. So in each lobe we said that 2 theca is present. In 2 theca we have pollen sacs, which is referred to as microsporangia, and they will be having pollen grains. Very simple, right? Yeah. Bilobed nature is very distinct. So you can just, if you put them under very close to your eyes, you can see there are 2 lobes, and that too in transverse section. Transverse section is like, I have this, I cut it like this. This is transverse, and when I cut like this is longitudinal. So I am going for a transverse section, and bilobed is very clear, even tetragonal is very clear, and the sporangia will be present at the corners. Microsporangia will develop and it will form pollen sacs. So just imagine like this. Okay, so microsporangia 1, 2, 3 and 4. So microsporangia will be formed only at the corners. This microsporangia will then be formed into pollen sacs, and inside this we have pollen grain. Okay, hope you are understanding. Extend longitudinally all through the length of the anther and are packed with pollen grains. So it just goes like this on the bottom. Okay, everything is filled with pollen grains, right? Now let's look at the diagram. So this is a transverse section. I am taking an anther and I am just cutting it this way, okay, horizontally. Okay, I'll just write it here. Transverse means horizontally, then you remember this because we will be using this interchangeably. Vertical. Okay, so transverse is horizontal, longitudinal is vertical. Okay, now just look at, this is the complete anther. Let me put a line here. Now it is bilobe, lobe 1, lobe 2, and let me put a box. Theca 1, theca 2. Okay, just imagine this is anther, left lobe, right lobe, and one lobe we have one theca, another theca. And look at this microsporangia, it is towards the corner. Microsporangia is towards the corner. Okay, so see, microsporangia is towards the corner. Okay, so microsporangia is towards the corner, and remember one more thing, that they are surrounded by layers 1, 2, 3, 4. Okay, and they are very different in their structure and they have very unique properties. Let's come from the outer type. Epidermis, most of the structures of plant has the outermost layer as epidermis. Then we have endothecium, epidermis, endothecium, then middle layers. So 3 layers together, they have protective function. They protect their pollen grains. Okay, epidermis, endothecium, middle layers. All these things play a major role in protection. And then we have the innermost layer. Okay, this has nourishment function. Okay, so you can see the top 3 layers, epidermis, endothecium, middle layers. All these layers have protective function. The middle layer is nourishment function. Just remember that. Okay, and this tapetum, when you see, they have dense cytoplasm. Remember, wherever you find something which plays a role in nourishment, they will have dense cytoplasm and they will have 2 nucleus. So having 2 nucleus may not be the case always, but yes, dense cytoplasm always happens. Think about how do they have 2 nucleus for a while. I'll explain it later, but then still you can think about how do they have, or how did they get 2 nucleus. Just think for a while. Okay, so when I say microsporangium, this is the microsporangium. Anther is the overall structure. Anther has 2 lobes, tica is 5. When you take a TS, when you look at this sporangium, so this is microsporangium. Okay, this is a sporogenous tissue. Inside the microsporangium, we find the sporogenous tissue. So that microsporangium structure we are studying. Okay, see here, it looks like a butterfly, right? Let's assume this to be its body, and they also call this as butterfly shaped because it looks like a butterfly. And then you see here, during the process of dehiscence, this epidermis, endothelium, middle layer, these stuffs break, and it gives a way out for pollen grains. Okay, and you can see one more thing called connective. This connects both lobes together. Fine. Are you able to understand this diagram? I hope you are able to understand. Yeah, just understand one thing. You have outermost layer epidermis, endothelium, middle layers. 3 of these layers are involved in the protective function. Tapetum is involved in nutrition, and when you come inside tapetum, you have microsporangia. Within microsporangia, you have sporogenous tissue, they have mentioned. This sporogenous tissue is basically the microspore mother cells. They are involved in the formation of microspores. Okay, I hope you are able to understand, and these terminology is also important. If I say transverse, it is horizontal. If I say vertical or longitudinal, that is same. Okay, just remember that. I hope letters are clear. That's good. Okay, so let's move to the next slide. Okay. Okay. So in transverse section, okay, in horizontal section, typical microsporangium appears near circular in outline. So we spoke that they are tetragonal and now you are circular. Don't get confused with the shapes. Let me make it clear to you. Okay. So the anther as a whole, so this box, the anther as a whole is obviously tetragonal. Okay, the box as a whole is tetragonal, or I will say it is like butterfly shape because that's how it looks. Okay, this stuff. But if you just look at the microsporangia, so this is distorted, but let's look in this diagram. If you just look, it is near circular. Though it's not like, see here it's a kind of oval, it changes, but it's near circular. This is the microsporangia. Okay, when I say shape of the androecium, that goes to be, when I say shape of androecium, what you should say? Shape of androecium is, come on, shape of androecium is tetragonal, and the shape of microsporangium, okay, microsporangium is a place where pollen grains are made. So that is actually, it's not tetragonal, it's circular in outline. Oh, sorry, circular in outline. Yeah. And then we saw this, they'll be having 4 wall layers. What are they? Epidermis, endothelium, middle layers, tapetum, and there will be outer 3 layers which perform the protective function, and they will dehiscence, and after they dehisce, what will happen? Okay, so once they undergo dehiscence process, pollen grains will be released. Okay, then innermost wall layer is known as tapetum. So remember how many wall layers are there? 4. We can group it into 3 and 1. 3 is epidermis, endodermis and middle layers. Ependyme, okay? Ependyme, epidermis, endodermis, middle layer. So we have 3 layers which helps in protection function, later dehiscence, and it helps in the liberation of whom? Pollen grains. When I say the innermost layer, the 4th layer, that is actually the tapetum. Tapetum gives only nourishment. So what happens as the pollen grains grow, it uses up the tapetum. Okay. Cells of tapetum, how will the cells of tapetum be? We just look, no, how will it be? It will be having dense cytoplasm because more and more nourishment will be provided if the cytoplasm is dense. And also remember, it has more than one nucleus. It has 2 nucleus. How does it have 2 nucleus? Can you think of how epithelial cells could become binucleate? What is binucleate? 2 nucleus, right? How? It is by a process known as endomitosis. Let me explain it here. Endomitosis. Let me explain. What is endomitosis? What is endo? This is within. Mitosis is equational division. Okay, equal division, right? So let me say we have a cell. Fine. Then we have a cell and a nucleus. So what usually happens is the cell's nucleus will double up and then the cell will divide. It will result in 2 separate cells. This is normal mitosis. But what happens in endomitosis? The cell will have, say, stage 1, its cytoplasmic content will increase. In stage 2, its nucleus will double, but it will not enter into the 3rd phase. It will be stopped here. So, how many nucleus does it have? 2 nucleus, and since more cytoplasm is formed, it will be having more cytoplasm. So, it undergoes a process known as endomitosis, so that a cell can have double the cytoplasm and double the nucleus, and it doesn't split up. Okay, so that's how tapetal cells become binucleate by process known as endomitosis. But there are some other theories also. This one is like commonly accepted, but there are some other theories also. When anther is young, a group of compactly arranged homogeneous cells, this is known as sporogenous cells. Okay, so in the center we saw the sporogenous cells. Sporogenous cells will be there, which then later becomes microspore mother cell. Fine. Sporogenous tissue occupies the center of microsporangium. So the lobes the complete structure is anther. It has 4 layers and then round structures. It is called as microsporangium. Inside microsporangium, we have sporogenous cells, which later become microspore mother cell. Okay, is that clear? I hope the structure of male reproductive system is very clear to you without any doubts. The next video we will see the microsporogenesis and the associated concepts. Hope this video is clear. If you have any doubts, please feel free to put it in the comment section. I'll answer to your queries. Happy learning. See you in the next video. Bye.